Okay, welcome to part two of the nervous system lecture. We're going to get more into the function. We do have a little bit of structure to talk about, but more about how we get the job done in the neurons. So I've got some sort of preliminary questions here for part two. What ions make up the resting membrane potential gradient? Okay. You can see them right here. Here's one here. There's one there. Okay. You can see the colors. All right. You can see where they're located. What is the most important resting membrane potential ion? All right. We'll get to that in a little bit. What do we mean by refractory period? I did mention that in the last lecture, but we'll talk about it some more. We actually have two different refractory periods. Okay, got the absolute and the relative refractory periods. Well, what does refractory period mean? All right. How do we stop neurotransmitter signaling? Because once we start it, we got to have a way to stop it, right? Just important to exhale as inhale. Just CO2 is just as important as oxygen when it comes to life. All right. Got to learn how to relax a muscle rather than just contract. So, how do we stop neurotransmitter signaling? And you should know sort of three and a half pieces of that puzzle. Well, and we'll get there. That's what we're going to work on this time around. Now, this slide says things to know. Well, yes, I would also say terms to know. We need to know the concentration gradient and electrical gradients for sodium and potassium. Hey, there's the answer to one of those questions. Sodium ions and potassium ions cross the membrane. If these concentration and electrical, we call that an electrochemical gradient. If these gradients change, that changes the voltage. It says we need a semi-permeable membrane. Means that a semi-permeable means it lets some things and doesn't let others. I like to say permissible rather than permeable. I do that because, again, what are we allowing across the membrane and what we aren't? Some things we allow, some things we don't. And that's partly how we make these electrochemical gradients. Electro means charge difference, all right? That's potential energy, all right? Like ions will repel each other, so a negative and a negative will repel, and a negative will be attracted to a positive ionic charge. All right, and we do get a difference in the electrical potential across the membrane. Chemical means that there is something dissolved, some kind of something. It could be an ion like chloride or sodium. It could be a phosphate. This is a chemical or an atom, okay, is also a chemical. All right. And in the second sentence here, we say temperature, concentration, charge, and size influences its ability to get across that membrane. All right. If you heat up the membrane, it'll become slightly different in terms of its permeability, or you cool it down. How much is present on one side or the other? The concentration difference. How much charge that chemical, that ion or molecule has, and how big it is. All of these things influence how or whether it gets across this membrane. And again, if you have differences and they have charge, you're going to have not only a chemical gradient, you're also going to have electrical, which is why we call it electrochemical. Okay, now let's talk about this term, resting membrane potential. Most cells, and neurons are no exception to this, are slightly more negative inside the cell voltage-wise than outside. All right? Have slightly more negative charge inside than outside, and the magic number is minus 70 millivolts, so slightly more negative. And so we need a way to sustain that and restore it when it changes, because the changes are the fundamental of signaling. So we maintain it by a protein called the sodium-potassium pump. It pumps sodiums out, which are positively charged, 3 of them, and it pumps also positive charge, but a different ion, potassium, in to the tune of 2. So for every molecule of ATP, you pump 3 sodiums out and 2 potassiums in. You metabolize 150 pounds of ATP every 24 hours, which means like 7-8 pounds an hour. All right, that's amazing. And most of that energy is used to sustain or restore the resting membrane potential. So you're pumping this, and this establishes gradients. You got lots of sodium on the outside, lots of potassium on the inside. Well, how do we get this difference? Since we're moving positives and negatives, that's where the leak channels come in. And we have leak channels that let sodium back in at a certain rate, and we have leak channels that let potassium out. They're moving down their chemical gradients. All right. Sodium wants to move in by virtue of its diffusion gradient. Never mind that it's a little bit negative in here. And potassium wants to leave because of its diffusion gradient. And potassium's the big one. Potassium's the important one. We have a lot more potassium channels, leak channels, than we do sodium. And so potassium leaks out faster than the sodium gets in, and that's why we're sitting at minus 70. You also have some negatively charged proteins. Yeah, I mean, a lot of the biomolecules, amino acids, et cetera, have a negative charge. So there are biomolecules that contribute to this negative charge as well. Before we go on any further, and I don't mean to interrupt the lecture, I don't mean to kind of take you aside. I think it would be a really, really good idea if you go into my Ivy and go to our class section. I'm going to try to upload this in this video. You can also find it on Netflix. It's look up resting membrane potential, but include the word Pearson also. So put in Pearson resting membrane potential. You can find it there if I can't upload it. But watch this video. I really, really like it. It's very direct and simple. It's very well done, very professional, worth your time. Okay. It shows you how the resting membrane potential is maintained and established, and even shows you what happens if you start changing the permeability of these ions. Okay. Worth it. So the electrical signals that neurons send are dependent on how the ions move or sometimes don't move, all right? When we have resting membrane potential, we're certainly pumping sodiums out and potassiums in, and you can see the darker purples here. That's because we have more potassium leak channels than sodium channels. So the potassium is leaking out faster than the sodium is leaking in, and that gives us a net negative charge. All right. Now, if we add some other channels in here, and they're called gated channels, we can change the rate of whether or not these move at all. All right, you can slow them down, you can speed them up, and the more you do that, the more you alter the voltage across the membrane, and that's how we signal. All right, this, by the way, is just another picture. It's showing you the sodium potassium pump here. It's showing you the potassium link channel sodium. It's a nice picture, but doesn't show you the ratio, so to speak, of many more potassium channels and sodium channels. So it's resting membrane potential is primarily determined by potassium, but if you start changing the rate at which potassium moves, you can change the voltage. You start changing the rate that sodium moves, you can change the voltage. And chloride, okay, chloride's a negative one. Remember how we talked about anesthetics, right? The general anesthetics, what they'll do is they'll let chloride in, make this super negative. And when you get super negative, that's called being hyperpolarized. And when you're hyperpolarized, you're not sending off action potentials, all right? Very cool. If you're not sending off action potentials, you're not having thoughts, you're not sending signals. That neuron is depressed, okay? That way that we vary the rate of ions moving across that membrane are what are called gated channels. These are channels that can open and close based on the conditions or the presence or absence of certain factors. For instance, if you press your finger on the skin and you activate a Pacinian corpuscle or something, that is a mechanically gated receptor. And what you've done is you have caused an opening of primarily sodium channels. The sodium floods in and it depolarizes the cell, and we fire off an action potential. Many channels are chemically gated, are neurotransmitter receptors, for instance. The neurotransmitters are chemicals, And if the chemical is present, the channel will either open or close, depending on what kind of a channel it is. It's gated, and we have as many that close as open. It will cause that channel to change configuration, the presence or absence of what we call a ligand, but it's a chemical. All right. And we have channels. We'll talk about these. We have some calcium channels that are voltage gated, means they open and close depending on the potential difference across the membrane. Now, this is the original slide. It shows you the steps involved in what is known as an action potential, which is the primary long-distance signaling mechanism down an axon. Okay. When you reach what is known as threshold, and that is a certain voltage in the axon hillock, you get a drastic change in permeability, first of sodium causing a depolarization, then of potassium causing repolarization. And that's what we're showing here. We're at resting membrane potential here, so that's step one. And we get some kind of a signal that causes the sodium channels to open and sodium floods in. And so the voltage goes way up because sodiums are positive. You let positives in, that's going to cause the voltage to rise. That's why we call it the rising phase. Then at a certain point, about plus 30, those sodium channels close. So they open at minus 55 and they close at plus 30. Kind of cool. It's double gated. All right. It's got two gates. One that opens at minus 55 and one that closes at plus 30. Then around plus 30, we have some potassium channels, and those open, and that lets potassium fly out. So we're letting these positives come out of the cell, which means the voltage starts dropping again precipitously. That's called the falling phase. That's shown right here. So now we've got potassium flowing out. We've got the repolarization phase, okay, the falling phase. And then there's this point where we have to reset the channels. In fact, we fall so fast, so quickly, we actually get below. We actually get to minus 75, minus 80 millivolts. We are hyperpolarized. And it's during that hyperpolarization that we reset these channels back to their original configurations, get them ready to fire off again. So we're using gated channels. These things are voltage gated, which means they open and close depending on the membrane voltage. All right. And once they fire off at threshold, starting with the sodiums and then the potassiums, it's what we call an all or none. We're going to go all the way up to plus 30, and we're going to come all the way back down. It's all or none. We're either going to fire off an action potential or we aren't. If we don't hit threshold, we don't fire off. We don't get half an action potential. We always get a full action potential. All right. That's what we mean by all or none. Well, here are the steps in a little more detail. So we got time on the bottom. All right, we've got voltage on the y-axis. Now notice where this probe is. It's in one little spot. It's in the node of Ranvier, or you could have put it right here, or you could have put it right here. Or you could have put it right here too, or right there. All right, could have put it anywhere where there isn't any myelin on that axon. Okay, that's where these action potentials occur, on unmyelinated sections of axon. If the axon's completely unmyelinated, it moves as a wave. If the axon is myelinated, it moves saltatory. You get this sort of periodic, you get this boom, 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 boom kind of thing. All right, like semaphore in France, ancient France. Okay, fire off, fire off, fire off, fire off. Okay, but we'll get to that in a little bit later. Let's go through these steps. I've animated this a little bit. So here at 1, that's our resting membrane potential. See, we're just sitting here waiting for a stimulus. And at 2, we get some kind of stimulus. Usually this stimulus at 2, between 2 and 3, all right, right there, that's caused by depolarization on an earlier segment of the axon. Okay, in this spot right here, what's happened is we have an action potential that fires here, and there's enough signal crossing that myelin sheath that it causes the voltage to rise just by virtue of depolarizing up here. Okay. Now, how does this one fire off? That's the sum. It's the summation of all of the input signals coming in. If the sum of all of those signals reaches our threshold voltage, we're going to fire off. So this signal here at the axon hillock is caused by the dendrites and other signals coming in. This one right here is caused by the previous segment of axon. And so this one here will stimulate the next segment and so forth and so forth. I'll show you some graphics about that. So we get that stimulus, all right? We hit threshold minus 55, and that causes the sodium channels, the voltage-gated sodium channels, to open up. We get increased permeability of sodium, and so the voltage rises in a fraction of a millisecond. This is 1 millisecond, 1/1000th of a second, and we go from minus 55 to plus 30, a swing of 85 millivolts, okay? Big swing. When we hit that plus 30, those voltage-gated sodium channels close. That's why we have a down arrow. The P stands for permeability or permissibility. The permeability rises as we let sodium in. Then when we hit plus 30, those channels shut, and the permeability to sodium drops to basically zero, okay? At that same point, we open up the potassium channels, and we let that potassium out. That's the falling phase. That's the repolarization phase. Again, you see the arrow pointed up and the P, that's permeability for potassium. So we're letting potassium out of the cell. That causes voltage to plummet even more than the sodium caused it to rise, and we get that repolarization phase. And it goes actually past minus 70. It actually gets down into minus 80 or minus 85 in some cases. That's the hyperpolarization phase. We are hyperpolarized until we get back to minus 70. It is during this time that those potassium channels shut and the sodium channels reset. It's important for me to say it that way. The potassium channels shut and the sodium channels reset. And so as they shut and reset, we reestablish resting membrane potential and we're ready for another signal. But that's not the end of the story. We don't have to get back to resting membrane potential in order to fire off again. And then that's very, very important in terms of how we send information down these neurons. It's not that we send just an action potential, but it's how many and how frequently. All right. And so we have to vary the frequency that we fire off these action potentials. Right about here, right? A little bit after 1 millisecond. Sometimes you see it at 2 milliseconds. It doesn't matter all that much. But somewhere out here, enough channels have reset. Enough of those sodium channels have reset that if you get enough signal to overcome that hyperpolarization, you'll fire off again. Okay, it takes a stronger signal somewhere starting here around 1.5, maybe 2 milliseconds. That a stronger than normal depolarization stimulus can get us to fire off another action potential. If it was just a barely minimal stimulus that just barely gets us to threshold, we'd have to wait until it was out here, much longer. We could fire it off between 1 and 2 milliseconds if it's a strong signal, but if it's a weak signal, we're going to wait until maybe 4 or so, okay? So we get a profound difference in the abundance of action potentials depending on how strong the depolarizing signal is. From here, from when we hit threshold to about this point here at 7, okay, where we start resetting those sodium channels, this is known as the absolute refractory period. There is nothing that's going to cause this to fire off again. Remember, it's all or none. We're only going to go this high. We're never going to go higher. We're never going to go lower. All right? We go up to here. We come back down to here. That's the all or none principle. But between that 7 and somewhere around here, around 4 milliseconds, between here and here, this is known as the relative. And another word for relative is maybe. Okay, it's all relative. It depends on what's going on. We have a stronger signal that will overcome that hyperpolarization. We get back to minus 55, even though we're hyperpolarized. It gets us back here, we're going to fire off another signal. If it's strong enough to overcome that hyperpolarization, you'll get a signal. You'll get an action potential. And that's basically what's shown in this figure. You see here in the light green, here's our rising phase, here's our falling phase. That's potassium leaving. See, somewhere a little bit after 1 millisecond, we go from this green to blue, dark blue, okay? Light blue, we can fire off if we get a normal signal, okay? Dark blue, a normal signal is not going to do it. It's going to be sub-threshold. That's the term, okay? Here, it won't matter how strong the signal is. We're already in motion. Okay, so it's absolute refractory period. But out here, where we're resetting those sodium channels, we get a strong enough signal, it'll fire off again, which means we can fire off a signal every about 1.5 to 2 milliseconds. If we have a strong signal, it takes at least around 4 or maybe 5 milliseconds if it's a weak signal. And that is important in the information we're sending. The faster and more abundant the action potentials are, the more neurotransmitter we're going to secrete. Now, I'm including this. I'm not really going to talk about this, but it should make some sense if you look at it. It's showing permeability and voltage. Here are the channels, okay? So we get a stimulus, we get the sodium channels opening, letting sodium in, the voltage rises. We close the sodium channels, we open up the potassium, the voltage falls. It's just showing you the channel configurations here, all right? This is what I want to talk about. So when we're at resting membrane potential here, you see that? Between resting membrane potential and threshold, we have a high degree of excitability. This neuron is ready to fire. So if we just get that 15 millivolts, just enough to displace it to minus 55, boom, we fire off. But as soon as we hit 55, that excitability, wham, goes to 0. This is when it excited means that we can't fire off another action potential. That's absolute refractory period. Now here, this figure is showing it at 2 milliseconds. In the other figures, it's showing it a little sooner. But as we hit that relative refractory period, this requires a really strong signal. Maybe not quite so strong, maybe not quite so strong. And when we get right back here, we're back at high again. So back to a relatively weak or normal signal will do it. Anywhere from here down, it takes a stronger and stronger signal. Here, it won't matter how strong it is. That's absolute. That's relative refractory period. So here's the narrative. Here it is in verbal form with no pictures. Just listen, take a note or two. We need some kind of stimulus that raises the voltage above that minus 55, that threshold, if you will. We have to cross the threshold to start that rising phase. And that happens at the axon hillock, okay? That depolarization will trigger a voltage change downstream, and it'll trigger the next action potential, the next patch of membrane. But that depolarization is due to sodium flowing in. That's positively charged, causes the voltage to rise. Now, the potassium channels will catch up, and they open up slower. They open up mostly about the time we hit plus 30, okay? You don't have to worry about exactly when those channels open, but just understand they're fully open by the time we hit plus 30. The sodium channels close right at plus 30. Right at plus 30, they get inactivated. And now those potassium channels, those slow pokers, are now in full effect, and now we're letting potassium flow out. And now we are repolarizing. We're in the falling phase of the action potential. So much that we shoot by that resting membrane value, minus 70. Close those channels. We're in hyperpolarization. The channels are resetting, the sodium channels in particular. All right. We don't want to get into it too much, but sodium channels have two gates, okay? One that opens initially and the other one that closes, and you have to reset them. And that's why in the hyperpolarization phase, we have that relative refractory period as those channels reset. Okay? And once we get full reset, assuming we haven't had another strong signal come through there, we get back to resting membrane potential. It requires that sodium potassium pump. It requires those leak channels doing their thing. Now, I wish this slide was a little better annotated, so I'm going to talk through some of this. If we are up in the dendrites or we are in the cell body, if you get a depolarizing or a hyperpolarizing stimulus, it doesn't matter which, you're getting a voltage change. Okay, that's depicted by the width of that white line there. And as that voltage change manifests across the membrane, it's kind of like when you throw a rock in a smooth pond. You see a big wave at the beginning, right where the rock hit, but then that wave spreads, okay? And as it spreads, those waves aren't as tall as they were initially, and they weaken and weaken. So the further this stimulus has to travel through the neuron, the more that voltage displacement diminishes. All right. That's due to the fact that we're still pumping ions. Okay. There's a little bit of intrinsic resistance in the cell itself. And so that voltage displacement will weaken. So if we get a stimulus way out here, it's probably going to require several stimuli in order to be strong enough when it reaches the axon hillock. That's what we mean by summation. Or if we get a negative signal from somewhere else, and that will cancel out the voltage, and we never reach action potential threshold. Okay, this is the premise of a graded potential. Okay, we aren't really talking about that one much, but I wanted you to understand what this figure really says. But if we get into action potentials, what happens is you get a displacement, and then the next patch of membrane feels that displacement and it fires off. And the next patch, it fires off. It's like a set of dominoes. This domino falls, which then causes this domino to fall, and this domino, and this domino. So what do you see? You see a lot of displacement. It's weakening a little bit, okay? The physics of depolarization doesn't change from a graded potential to an action potential, but we're ready to fire off if you get that displacement. So it self-reinforces. That's why you can send these axons meters. You know, cells are 1/1,000,000th of a meter, and you can send it. It's like sending it from here to Paris. All right. And it doesn't weaken because it self-reinforces. We call that a wave, a self-propagating wave. It's like a set of dominoes. Okay. With the myelin sheath, it just speeds that process up. That insulator prevents the weakening of this signal, okay? And this signal moves pretty fast, okay? Faster than if it's unmyelinated, okay? So the wave propagation, if you will, where you get this sort of domino, 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 domino on an unmyelinated axon, we can get 2 meters per second. So like, let's say from the top of my head to the bottom of my toes in about a second, okay? With the myelinated, that rate, it goes up big time. 100 meters per second, almost instantaneously, faster than you can perceive it in some cases. Okay? Which is why if you put your hand on a hot stove, man, your hand is practically behind you before you even realize what happened. All right? This is called saltatory conduction because the depolarization jumps from node to node. That's what saltatory means. Here it's more continuous. It's like a set of dominoes set up. The voltage here causes the action potential here, causes the action potential here, et cetera, et cetera, et cetera. It's doing the same thing here, but it's traveling so much further, a full millimeter for every myelin segment, that it speeds it up 50-fold. Pretty cool stuff. A little mental break for a second. I saw this slide. You don't have to be crazy. Just hang out with me. I'll train you how to be crazy. Get crazy about these neurons because you can do so many things if you understand how neurons do their thing. You can understand why we give somebody an antidepressant, how a certain drug works to cause anesthesia, why it's local as opposed to systemic, general, why certain pain relievers do what they do and others do what they do, et cetera, et cetera. Opioids. And it's a huge world. Buckle up because it's all about neurons. So I've shown you that action potential, and now the next piece of the puzzle is how do we get this action potential to transmit to a downstream cell? Whether it's another neuron, or it's a muscle, or a gland, we still have to get this electrical signal to this cell, which is not connected to this original neuron. All right. It's showing you the mechanism here, and I'll kind of briefly go through it. You get this action potential, and it's coming down both sides here. Okay, it's like a circle around this, a wave passing through here of depolarization. These channels here, and this is a cross section of a channel, okay, they are sensitive to voltage. It's an ion channel for calcium. So as this wave comes in, these channels open, let calcium, and then calcium turns on enzymes that cause these vesicles to merge with the plasma membrane and release that neurotransmitter. Now these channels over here are sensitive to that neurotransmitter. They are chemically gated ion channels. And so when they see that neurotransmitter, they'll change configuration and let in an ion and cause a voltage change, usually, not always, but most of the time. If it's a muscle, sure, let sodium in and start depolarizing. We get an action potential down here, okay? In a gland, maybe we let calcium in and we get contraction, okay? Secrete saliva in some cases, all right? There are some rare synapses, and I'm sighing a little bit here, where you have, instead of this space, you have gap junctions. So you let calcium in here, and then the calcium can move over to the other cell. Those are known as electrical synapses, super duper fast, really important in cardiac muscle. You do see some electrical synapses in super fast responses like the blink reflex in some cases. But the vast, vast majority of synapses are chemical. So here's just showing that same figure again. Here's the presynaptic terminal, sometimes called a terminal bouton. It's the French for button. Okay, it's kind of mushroom shaped in 3 dimensions. Here comes that action potential down here. Causes these voltage-gated calcium channels to open. Calcium diffuses in, serves as a cofactor to cause exocytosis on these docking proteins. That's a little too complicated, I think. The calcium comes in, triggers exocytosis. Now we've got neurotransmitter in the synapse, and if there are receptors, those receptors, again, they can be chemically gated voltage channels or other types of receptors. They will then trigger a response in that postsynaptic cell. Let's take a look at some pictures. This is an electron microscope. Here is one of those terminal boutons, and these are granules of neurotransmitter, okay? So what happens is the action potential comes down, causes calcium in, and then those granules are forced to fuse with that plasma membrane. Look how small that gap is. It's so small, it's barely larger than one of these granules. It means the diffusion is exceptionally fast, all right? Here are the receptors would be on this surface. This happens to be a muscle cell, okay? It's a motor end plate, all right? Okay, so those granules would be acetylcholine in this case. Those granules open, release acetylcholine on those receptors, and off we go. Here's a little bit of a picture on the right. It's showing you the terminal bouton here. It's showing you the dendrite, a downstream neuron. You can see that synapse is so small. This is the cartoon that's kind of enhancing that picture. You can see some granules here. Oh, there's a mitochondria in there. We aren't talking about mitochondria that much. They're important for recycling acetylcholine. But in any case, here are those vesicles. And again, look how small that distance is. I mean, the diffusion is almost instantaneous. My animated video from my class. So we get this action potential, spreads down across through here, causes that voltage displacement. You've got voltage-gated calcium channels. All right. In another life, you'd call those L-type calcium channels. All right. They open up, and calcium is higher outside the cell than inside the cell. So the calcium, these guys open, they facilitate diffusion of calcium into the presynaptic terminal. That calcium then facilitates exocytosis. There are those docking proteins, right? All right. And we get diffusion, okay, of neurotransmitter into the synapse. And if that cell has receptors, it doesn't always. Sometimes it gets those problems, okay? Sometimes we have autoimmune disorders and things, all right? They will bind, some of those molecules will bind the receptor, triggering a response, okay? It could be muscular contraction. It could be some other cellular change in behavior, okay? Or maybe we trigger a voltage change, and now we're going to continue that signal down another neuron. All right, that's how we get the signal transferred from one cell to another. But that's not the end of the story. Sorry. So here's the verbal equivalent. I'll just go through this quickly. I've already said it a few times. If you don't know it by now, it's time to write this stuff down. Okay? The action potential arrives at that presynaptic terminal, that bouton. Triggers voltage-gated calcium channels, and so calcium's higher outside the cell in concentration. The calcium diffuses in, causes these vesicles to fuse with the membrane. Exocytosis, yes, it requires ATP. Okay, fine. All right. You get that exocytosis, and you have release of neurotransmitter and diffusion across that super short distance. Okay. Binds to receptors. The receptors can determine the type of response that you get. It could be a voltage change. It could be a behavioral change, all sorts of things. All right. That's the postsynaptic membrane. If it's a neuron or maybe a muscle, it'll initiate what we call a graded potential. I've mentioned that phrase before. So you're getting a voltage displacement on the downstream cell. That could be a muscle cell. Again, another neuron. If it's excitatory, it's causing depolarization, excitatory postsynaptic potential. Or if, say, it's letting chloride in, okay, let's letting chloride in. That makes the voltage more negative, with less likely to trigger an action potential, inhibitory postsynaptic potential, okay? Depends on the neurotransmitter, okay? We talked about that in class last night. Depends on the kind of channel, what the ion is that's moving across that membrane. Step 6 is where we begin shutting the synapse down. We don't want that neurotransmitter sitting out there all day. Okay, generally the half-life of neurotransmitter is measured in fractions of a millisecond. And you guys know what half-life is, right? If not, ask me, okay? One of the things that happens is we have to shut down the release of calcium on that presynaptic terminal. And so you have receptors on that presynaptic terminal membrane that will shut down calcium release. Okay, you got to stop the calcium or you're going to have exocytosis. So you're going to export that calcium, sequester it, do whatever you have to prevent that calcium from being around. Then you're going to need to get rid of the neurotransmitter that's been released. And we have 3 mechanisms. One is reuptake. We bring it back into the presynaptic terminal. Okay. Another is an enzyme that's found inside the synapse. It's not inside the cell. It's inside the synapse, so it's outside the cell. That will chew up and destroy the neurotransmitter, degradation. Or remember that the synapse is extracellular. It is extracellular space. You create a concentration of neurotransmitter, it's going to diffuse away. So 6 and 7 are how we stop the neurotransmitter. And like I said, we starting is important, but also stopping is important. Okay? Sometimes we're going to want to slow down the stop for certain types of antidepressants and so forth. Okay, there are clinical reasons for doing that. But most of the time we want to shut those down pronto. And I'm going to show you that in a video here in an animated slide. Here's the animation. We've got that calcium coming in because we've got that action potential coming along. Okay. We got it first, and we've also got the neurotransmitter out here. So we've got to deal with the calcium, and we've got to deal with that neurotransmitter. As long as neurotransmitter is present and bound to a receptor out here, we will get a response. Okay. Maybe that response is wanted if we want to sustain a signal, like for instance, we want to continue contraction of a skeletal muscle. Sometimes we want fast termination, like when you're blinking your eyes or something along those lines, or when you're listening to a sound, for instance. So we've got to get rid of that neurotransmitter. So first we got to get rid of that calcium. So we saw the calcium go away there. Got to shut that down. And that's caused by neurotransmitters binding the surface of the presynaptic terminal. But we also have to get rid of those. And I told you about that in the previous slide. The first mechanism is called reuptake. And the presynaptic terminal can do this. A glial cell or a microglial cell can do this. All right. It is removing by transport the neurotransmitter from the synapse. So neurotransmitters can be returned by using reuptake. The second mechanism right here, you see this little blue circle? And then you might have blue circles all around in here. We got just one here, okay? You see how there's a red dot here and a bunch of little red dots there? What's happened is this is an enzyme that chews up, it metabolizes that neurotransmitter into an inactive form. This process right here is known as enzymatic degradation of neurotransmitter. The enzyme degrades the neurotransmitter, and so the concentration falls. Okay, we did talk about one of these. We mentioned it last night. We mentioned it in muscles, acetylcholinesterase. This is an acetylcholine molecule. This is acetylcholinesterase. We chop up the acetylcholine into acetate and choline. Okay, enzymatic degradation, super duper important for acetylcholine transmission. So the enzyme inactivates neurotransmitters through metabolism by chopping them up. There's our acetylcholinesterase, just to remind you. Our third mechanism, you just saw there with the arrows, is that this space is in between the cells. This is extracellular space. So that third mechanism, and show it there again, is diffusion out of the synaptic cleft. Okay? If you have a higher concentration here than out here, these molecules will diffuse out that way. And what's cool is this may mean that the neurotransmitters do double duty. Like for instance, you see this one right here, maybe this is acetylcholine, may trigger vasodilation. Okay, this neuron's been busy. It's been doing a lot of metabolism. It needs oxygen. It needs glucose. Well, we need to increase the supply of blood. So that diffusion not only removes neurotransmitter from the synapse, it also primes the blood vessels to bring more blood. That's what we talked about with those functional MRIs last night. So as long as we have neurotransmitter bound, we're going to have response. But as we diminish the concentration of neurotransmitter, less and less of these receptors are bound, which means less and less response. The lower the concentration of neurotransmitter, the weaker the response is. And if there's no binding, no response. Okay? So by diminishing the concentration of neurotransmitter, we terminate the response. So get rid of the calcium, reuptake, enzymatic degradation, diffusion leads to release of neurotransmitter off those receptors and termination of response. And so this concludes this part 2. We'll talk about it a little bit more during class. Bring your questions. Bring your questions. I love your questions. Thank you for your time. And I hope this was useful for you. I added a few extra slides relative to the PowerPoint that was in class. If you have any questions about those slides, please let me know, and I will be glad to answer your questions. Thanks for your time. Looking forward to seeing you tomorrow. Take care.